Okay, let's get started. Today we're going to be talking about this, the virus represented by this virion, so adenoviruses. Yeah, we'll finally get around to talking about these guys right at the end. I have a tentative yes for a guest lecturer for our last lecture from Luis Ruedas talking about virus ecology. I still need to bug him a little bit more. So with any luck, he'll show you gross pictures of hantaviruses and hantaviral disease. So it should be kind of fun. He's always a lot of fun. The main problem is cutting him off after the 2 hours of lecture that he gives. So, but first, of course, everyone's favorite are clicker questions for the day. The equivalent protein in papillomaviruses to the helicase domain of large T antigen is E1, E2, E3, E6, or E7. Five. So everyone thinks it's E1. Fortunately, I agree with you. What does E2 do? This transcriptional regulator. E6 and E7 involved in P53 and RB in terms of doing what? Cell cycle, regulating the cell cycle. So all of these double-stranded DNA viruses have to get the cells they infect to go into the cell cycle, because otherwise they're not going to be replicating their own genomes. And so that's the real critical aspect about these particular ones. We're going to talk a lot more about that in terms of adenovirus today as well. So our second clicker question for the day. The protein used in HPV vaccines is the major capsid protein, the minor capsid protein, E1, E2, or E7? Go for it. That's the point. You're supposed to be talking amongst yourselves. Five, four, three, two, one. I'm not sure we've ever had 100% before. Yay! So, point about HPV vaccines, yes, I heard you saying it. It's the L1 protein. L1 protein is the major capsid protein. Even if you didn't know that, why would it be a good guess to say the major capsid protein? What else is the immune system going to be recognizing? Exactly. And so one of the main points here, particularly with the HPV vaccine, is that it's a virus-like particle which has just that one protein in it. This works for some vaccines. Turns out for a lot of vaccines it doesn't. But it's probably because that is the one and only protein which is on the outside of both papillomaviruses, and the same thing is true for the polyomaviruses. The major protein which is present on the outside of these things. So let's select the right one so everybody gets points. Yes. Where was it? Green. Good. Show results. Moving on. So big picture with adenovirus. Wow, we cleared out today. Is there another midterm today? Tomorrow. Which class? Cell. That's why I see so many people in Rosensteel's office. Okay, that makes sense. So, yeah, maybe this will help a little bit with cell biology. So, big picture with adenoviruses. These are also double-stranded DNA viruses, but big difference from the papilloma and polyomas is they have linear genomes. So they're not double-stranded circular, they're double-stranded linear, and that immediately raises a problem whenever you have a linear genome, which has to do with replication and ends and how to deal with that. They're also a lot larger and more complicated than these viruses, have this really amazing structure, and to make this amazing structure they use lots of different proteins. The way they get lots of different proteins has to do with splicing, and as those of you in the molecular genetics class tomorrow are going to hear all about how splicing was discovered in studying the major late transcript of adenovirus. And then we'll talk a little bit about the obesity virus, and you can guess by the highlights here what I think about it. So a couple of key concepts again from the lecture today. Most of them have to do with structure. Again, this really amazing structure that we have for adenovirus, and literally in the last couple of years, and in fact since the textbook was published, there have been some really impressive structures of adenovirus, both X-ray crystallography and cryo-EM reconstructions that we'll take a look at in just a second here. That's really given us probably the highest resolution structure for the largest macromolecular complex that we have to date. Part of that's through difference imaging. Alternative splicing and alternative tailing, that's how you get so many different proteins that are important for putting the structure together. There's also important cell cycle regulation, which should be really familiar because we've already talked about this for the papilloma and the polyomaviruses. In terms of replication, has to do with terminal proteins. It should sound really familiar relative to which kinds of viruses? Way back when, last midterm? First thing we talked about before the last midterm? The polyomaviruses, not polio, but picoRNA viruses. See, eventually I'll remember these things. Polio, what does it have? Protein primed replication. So here, protein primed replication, very similar. Also has inverted terminal repeats. That's what ITR stands for. Also really critical in terms of the replication process. Where do these guys come from? Turns out they're not too bad in terms of disease, although strangely enough, the armed services is where one of the major issues has to do with some of these diseases. I love this micrograph because A, it's got these beautiful adenovirus virions in it, but it also has this little virus over here. What's that? The satellite virus, which hopefully George talked about. Adeno-associated virus. Remember the AAVs, the parvoviruses? That's one of them right there. Adeno-associated virus right here. This is sort of the classic picture with all of these projections at the five-fold axis of symmetry for adenovirus. So spend a while talking about the structure because the structure is just so absolutely cool. A little bit about binding and entry, but mostly about transcription and replication, and a little bit about how they get out. So where do you find these things originally? Found in respiratory and eye diseases. The originally isolated from adenoid tissues. That's why they're adenoviruses. A lot of people thought common cold, but what caused most of the common colds? Second most common, coronaviruses. Exactly. So they're not common cold, but there are certainly a number of respiratory and eye diseases. But the main reason that there's a chapter in the book about this is that they're really wonderful tools for understanding very basics about how transcription works. In fact, a lot of the early transcription studies were done on adenovirus. Also in terms of splicing. Again, splicing was originally discovered in adenovirus. First, people thought splicing was just present in viruses. And then, of course, we figured out that most of our genome is spliced as well. And more recently, adenoviruses have been very heavily used for gene therapy. In fact, one of the main vectors for gene therapy these days is the use of adenoviruses. There are a whole bunch of them, 100-odd adenoviruses, and that's what the book says. I think we're up to about 150 now. A whole bunch of different serotypes. What does serotype mean? Just talked about that in terms of the immune system for vaccines. Different antibodies. So all it is what you see, one antibody that'll bind to, one set of adenoviruses, another binds to another set of adenoviruses. And so these serotypes are literally what those antibodies actually bind to. There are some tumor viruses here as well, but just like we've talked about for all of the other DNA tumor viruses, only in non-permissive cells. They're not replicating virus, but they become transformed and grow uncontrollably. So we'll start out looking at the structure. This is, in fact, a slide that we've looked at before. Here is an older cryo-EM reconstruction of this virus particle, virion. 11 proteins go together here. If you've got 11 proteins in your virion, what does that mean about your T number? Has to be a pseudo. So that's what we put this T in front of it here. Pseudo T equals 25. How do we get 25? We've got to go from one 5-fold axis of symmetry to the next 5-fold axis of symmetry. This is really nice because it has these projections at each of the 5-fold axes of symmetry. And I'll turn on the pen and we'll take a quick look at that. So here is a 5-fold axis of symmetry. Let's make it red, a little bit easier to see. So here we have one 5-fold axis of symmetry, and we've got to get to the next 5-fold axis of symmetry. Here's one right here. Here's one of those projections. Okay, how do we get from here to there? We go 1, 2, 3, 4, 5. And we land here. If we like, we can even start here at this 5-fold axis of symmetry and go down to this one. Again, there's another projection coming out there at the 5-fold axis of symmetry. We go 1, 2, 3, 4, 5. So h squared plus hk plus plus k squared. K is 0, so it's just h squared, so it's going to be t equals 25. That's mostly made up of the hexon protein, 240 copies of that, and then 12 of your pentons. Not surprisingly, it's 5-fold symmetry. The penton is interesting because it's not just one protein, it's a whole bunch of proteins, 3 different proteins, in fact, that are there, the base, fiber, and nom. Most important for us is this hexon protein. This is the main thing that makes up that structure here. Everything is in between each of these 5-fold axes of symmetry. And we'll see when we look at the higher resolution structure in just a second, really nicely, that these are hexon proteins. So they go in each of these hexameric units, but they're actually a trimer. So they're 3 proteins put together. This had been solved through X-ray crystallography, just the protein by itself, many years before. And then this was the cryo-electron image that they had at the time. You put 3 of these together, red, white, and blue, nice and patriotic here, or bleu, blanc, rouge, depends on your point of view. Fit together nicely and make these pentons. We have these 3 projections coming off the top. The basis of these structure are 2 beta barrels. So this 2 beta barrel structure, also present in bacterial viruses and archaeoviruses. So we like to think this is an example of a really ancient virus fold structure. But this is only 4. So we've got hexon, base fiber and knob, but 11 proteins are present in the virion. Where are those other proteins, and how do you actually go about studying them? One way of doing that is taking advantage of the crystal structure of just this one protein. And so you take this crystal structure, you put it together like this, and then substitute this structure in the cryo-EM structure everywhere where you have a hexon. And then you see what's left. And then you try and figure out what those other proteins are. And that's what's called difference imaging. Here we have what each of those hexons would look like just by themselves. This is what the electron density looks like in your cryo-EM map, and you just subtract these guys from these guys, and you end up with these extra structures, which are sort of between each of the hexons. And now, again, with higher resolution imaging, we know exactly what those proteins are, but it's just a combination of the two techniques. X-ray crystallography for high resolution of one structural protein, and then lower resolution cryo-EM, and you can mix and match these two to see what things look like. Of course, that's great and wonderful until you get a better structure. And this is the structure from Science from almost 5 years ago now, 2010, where now they've done just a reconstruction based on electron microscopy of this virion. I think this kind of looks like something you should see in a candy shop rather than a virus particle. But each of the individual hexamers here in different colors, and then the pentons here at each of these corners, looking at this particular structure. Also, again, in a lot more detail. What was interesting is at the same time, in the same issue of Science, there was another publication. So one of them was an X-ray crystallography structure. The other one was a cryo-EM structure. And basically, they saw exactly the same thing here. Now, this is looking at where those sort of three-pronged structures were that we saw in the difference imaging before. Now you can actually see them in this cryo-EM reconstruction. It's these purple pieces, which we have right here. One of the things they really push in this paper is normally cryo-electron microscopy is a lower resolution technique, but here they were able to push the resolution to almost as good as X-ray crystallography. So if you look at, for instance, this arginine side chain here that they model into the structure, it matches almost perfectly with the electron density, which is this wireframe model here on the back side. So did want to take a quick look at some of these movies. I think I have them loaded up here already. Should hopefully show up. Yes, click, go through to them. Here we are, yeah. So these are those movies from that article. This is that structure. So you get to move. Actually see where all of these hexons are. The hexons, you can nicely see the threefold projecting structures that come out of each of those hexons, and then the proteins which are present in between each of these hexons, packing them together. You can also see nicely the fivefold axis of symmetry right here. And the hexons around it turns out to have a slightly different structure than the ones which are next to those as well. There was another movie to look at here. This is a structure now from the cryo-electron microscopy where you can literally see that there's enough resolution here to pull out where the peptide backbone is. Each of these is now, again, from taking, in this case, I think it was about 300,000 virus particles that they then all averaged together, and that's where they came up with this really ridiculously high resolution structure. And again, you can even see the connection down here at the bottom between the two different domains of this protein. This is one of the hexon subunits that then comes together as the trimer. Okay, so enough cool pictures. We'll move on and talk a little bit more about the structure here, mostly because this was a wonderful model system. Anybody know where this is? Anybody been there? Me? So this is Cold Spring Harbor Labs, sort of the center of molecular biology, certainly in the early days, 50s, 60s. This is where they had the bacteriophage course that Max Delbruck ran. Soon after that, they started to work on animal viruses, and this is one of the pieces of art they have at the Cold Spring Harbor Lab campus, actually on the top of this gazebo, looking out over Cold Spring Harbor on Long Island. One of the interesting things here is it turns out this is in fact a wastewater treatment plant that they put it on top of, which has nothing to do with adenovirus, but it's so iconic that they use it for that. The other thing which I really love about Cold Spring Harbor, they have a clock tower as well, but instead of the noon, 3 o'clock, 6 o'clock, 9 o'clock around it, they've got CAT&G around the outside. North, south, east, west, no, CAT&G. So what is in this really completely iconic structure? You've got genome on the inside. We'll look at that in just a second. But most importantly, what we've been looking at so far have been the hexons, which are these sort of rhomboid structures here, penton, base, fiber, and knob. And then the inside is the genome. Unlike the case for the papillomaviruses and the polyomaviruses, which we talked about the last couple of times, this is now bound by a viral protein. This is a viral DNA binding protein which is associated with your genome. And then, infamous red ovals here, terminal protein bound at each end of the genome. The numbers here are completely immaterial. I don't expect you to remember them, so don't bother with all of that. What does that genome look like? Again, it's got these terminal proteins bound at not both strands, but the 5' end of each of the 2 strands we have in the genome. It's depending on the adenovirus. Remember, there are at least 100 different ones of these, and between 30 and about 35,000 base pairs in length. Again, linear double-stranded DNA genome with a protein bound at the 5' end of this strand and of this strand over here. At the ends of the genome, you have 100 nucleotide inverted terminal repeats. And what inverted terminal repeat means is that if you go from 5' to 3' on one strand, CAT, CAT, CA, it's a typo in the textbook. The opposite strand, the other end, CAT, CAT, AC. What this means, of course, is that this end, as well as base pairing to its complement over here, could also be base pairing to this end over here. And we'll see that's absolutely critical in terms of the replication process. And again, as a reminder, don't always believe everything that you see in your textbook, because there are typos. Typos happen everywhere. Believe what I say, not what's in the book. So at least as far as the exams are concerned. And if you have a question about that, send me an email, like certain people who remain nameless do. And I will let you know. I'll go and look it up and figure out something other than the textbook in terms of what's actually going on there. So how do they get in? This is, again, just a reminder of all of the different kinds of entry processes that we've talked about. Forgot to show this last time, but simian virus 40, the polyomavirus, comes in through these caveolins, cavosomes, and then gets itself into the nucleus. Look at adenovirus. This is an interesting process. You remember, these are non-envelope viruses. It's just protein on the outside. So getting into, eventually, the nucleus is a lot harder than some of these enveloped viruses that just fuse at membranes. And when you fuse the membrane, then you release everything that's on the inside. So somehow these guys have got to get on the inside and then get their capsid dissociated. So what happens, they bind to specific receptor, Coxsackie, an adenovirus receptor, again, like polio. This was originally discovered because it was a receptor. I went and looked last night. We still don't know what this actually does in normal cells. That binds to, not surprisingly, the projections at all the 5-fold axis of symmetry. But then you have secondary interactions with the penton base, and we'll see a cartoon of that in the next slide. Then it goes into the endosome, a lot like you have with influenza viruses, a lot of these things receptor-mediated endocytosis. In the endosome, however, you can't fuse this membrane with the endosomal membrane because there's no membrane to fuse with. And so you degrade first some of the proteins on the outside, which is a viral protease, and then the capsid itself is released, and you can see capsid. It's in adenovirus-infected cells. They get transported to the nucleus, and then at the nucleus, this wonderful, beautiful icosahedral structure is too big to get through the nuclear pore. So it has to get degraded again at the nuclear membrane, and then you have the DNA, this linear double-stranded DNA, that gets released into the nucleus. And again, these are viral proteases that are involved in this. It's a very tough structure, these naked viruses on the outside. So we'll also see how useful that is a little bit later on. How does it get in? This is just a different cartoon from different source here. Here we have binding to the knob at the very end of these pentons, and then secondary binding with secondary receptors, a lot like what kinds of virus we talked about? HIV, co-receptors, you need both to actually get this in. Once you have association of both of these receptors and co-receptors, then you have normal receptor-mediated endocytosis. Again, receptor-mediated endocytosis gets into the endosome, released from the endosome, gets to the nucleus, releases the genome in the nucleus. What does that genome look like? It's crazily, ridiculously complicated. It's actually not that bad. Looks much worse than it really is. All of these lines, both the brown lines and the blue lines, represent transcripts coming from 8 promoters, 12 polyacytes, and then a couple of small RNAs as well. We'll walk through them here. First, the early promoters. So, early promoters, these are things that get turned on first. As soon as you get that DNA into the nucleus, what are early proteins usually involved in? Replication of the genome and regulation of the cell cycle, because just like the papilloma and polyomaviruses, these guys are infecting cells that are normally not necessarily replicating. So you've got to regulate the cell cycle in some way. You've got to be turning up what's going on present inside the cell. So we'll go through these individually here. The main ones are going to be E1A and E1B, because these are the ones that are regulating the cell cycle. And then the E2 proteins, which are DNA polymerases, terminal proteins, and DNA binding proteins. You also have all of these two, which are small RNAs. And everyone says, small RNAs, they only discovered these really recently. Actually, no, people knew about small RNAs way back when they were studying the adenovirus. Late proteins are all expressed from one promoter, the adenovirus major late promoter. A lot of times when you look at some of the older literature on transcriptional regulation, people talk about transcriptional regulation. They don't mean transcriptional regulation, they mean regulation of the adenovirus major late promoter, because this is pretty much always what was being used to look at that. This promoter has always the same sequences, or gives transcripts that always have the same sequences at the 5' end, but highly variable sequences that are present at the 3' end, generating basically all of those proteins that are going to be in the virions, each of these individual little pieces here. So if you look at the hexon protein, for instance, here, it's one of these late ones. Penton protein, that's the penton base. Here's the penton fiber. You also notice that these are all at different classes. These different classes, L1 through L5, are dependent on where you get polyadenylation taking place. So you've got differential splicing and differential polyadenylation that, again, we'll look at in more detail, and then a few more of these regulatory RNAs that we have here at the end. So early genes here, some small RNAs, the intermediate genes, the major late promoter, and then a couple of other small RNAs that are important for regulation of the cellular defense response. So what about splicing? Again, this was originally discovered in studies of adenovirus. This is a paper from, let's say the figure is from a paper by Phil Sharp, conveniently colored. The original is in black and white. It's way harder to figure out what's going on there. And basically at the same time, Rich Roberts and his group at where? Cold Spring Harbor Labs discovered basically the same thing. Phil Sharp at that point was at MIT. They used this process called an R loop, and that R loop is taking a messenger RNA and then hybridizing it to the DNA that you have in your genome. So in this case, they took the RNA that there was most of, surprise, surprise. It's for the hexon messenger RNA, because that's what you need the most protein of in order to put these full wonderful structures together. And then they hybridized it to, wasn't actually the whole genome, but a large part of the genome of adenovirus. And what they found was that this hexon messenger RNA hybridized to a nice long piece of DNA, which is this coding sequence here, and this funky little thing down here at the end. What would this be? Single-stranded. What do you get on messenger RNAs? Poly-A tails. So that's your poly-A tail. But the 5' end was what was the weirdest, because here, after you had this hybridization to the coding sequence for the hexon messenger RNA, there were these big loops, particularly this C loop, but also the B loop and the A loop. And those, they showed through very careful measurements in the electron microscope that this corresponded to this DNA that was non-coding, because it didn't hybridize to this final messenger RNA, next to some sequences that were present, the ones in black here, that were hybridizing, were making double strands. So this was the first evidence for splicing, that the DNA didn't code for the same thing as the RNA. So that was the major discovery here. For this, Phil Sharp and Rich Roberts both got the Nobel Prize. So splicing is not just cool and neat and you get the Nobel Prize for doing it. It turns out it's also really important, not surprisingly, for virus function as well. We'll start out with the first one, E1A. Gee, surprise, surprise. This is spliced, in fact, in 4 different ways. The only important ones, as far as we're concerned, are these two right here. N-terminus, a couple of conserved regions, CR1, CR2, and then CR3, which is missing in this spliced version, but present in this spliced version. I don't expect you to remember the numbers here, that's why I'm not talking about them. But they interact with some of our usual friends. PRB, RB protein. P300, which is what? Interacts with large T antigen, because everything interacts with large T antigen. It's a co-regulator of transcription, histone acetyltransferase. TBP, TATA binding protein. ATF is a transcriptional regulator here. And this tells you immediately that this longer protein interacts with the general transcription factors, whereas this one down here does not, but it's still interacting with RB and these transcriptional regulators. So what's this interaction with RB all about? Well, should hopefully be kind of a broken record by now. RB, normally cyclin-dependent kinases, gets phosphorylated, released from these E2F factors. Again, why are they called E2Fs? Because they're important for transcription of the early 2 genes in adenovirus, which is originally how they were found. How is that being regulated here? Slightly more detailed than we had when we talked about papilloma polyomaviruses. RB interacts with these histone deacetylase complexes, HDACs. Histone deacetylation takes acetyl groups off of histones. What does that do? Compacts the DNA. So you go to heterochromatin. Heterochromatin, you can't get all the transcriptional machinery into the promoters. You can't get transcriptional activation. So normal cell cycle, you phosphorylate RB, it falls off of your E2 factors, and you can activate
Most importantly, of course, the E2 genes in adenovirus, but normally S-phase genes as well. E1A interacts through that RB interaction domain, the conserved region, and basically does exactly the same thing as phosphorylation, releases these E2 factors, which are cellular factors, to go ahead and activate transcription for those E2 genes. Now, there's a couple of different E2 genes, but the ones we already talked about do what? They're DNA polymerases, they're terminal proteins, they're DNA binding proteins. They're important for DNA replication. So this is one of the things that E1A does. The other thing that E1A does is it regulates P53. So P53, again, guardian of the genome. We talked about it before in terms of activating the cell cycle. Normally, it would block progression through the cell cycle. In the absence of P53, you allow progression through the cell cycle. But at the same time, if you have lots of P53 around, it will also lead to apoptosis. And apoptosis is a really bad thing, again, to totally over-anthropomorphize if you're a virus, because it breaks down the cell before the virus has a chance to make a lot more of it. So how does that work in terms of E1A? E1A, the arrow here is supposed to be blocking P53 activation. I actually think the arrow should be more over here in terms of blocking the transcription of MDM2. Well, why do we care about MDM2? Normally, MDM2 binds to P53. That P53 gets taken out of the nucleus and broken down. So MDM2 basically keeps P53 at a nice low level, because if P53 gets too high, you're going to be blocking the progression through the cell cycle. You'll be leading to apoptosis. And in fact, that's normally how you regulate apoptosis, is by regulating MDM2, allowing a real increase in P53, which leads to arrest of the cell cycle and eventually apoptosis. So E1A will block P53 transcription of MDM2. No MDM2, you end up with more P53. If you have E1A also acts as an activator of transcription, because it interacts with P300 and CBP, which are the opposite of histone deacetylases. These are histone acetyltransferases that we talked about last time. Those put acetyl groups onto histones, which opens them up, makes them euchromatin, and activates transcription. This, among other things, activates transcription of a protein that binds to MDM2, which removes MDM2, leaves a lot of P53 around. Okay, so E1A is leading to a lot of P53, which is going to lead to apoptosis. This is bad for the virus. So what does it do? It brings in another set of proteins, the E1B protein, which basically suppress apoptosis. And they do these downstream of P53 or directly by interacting with P53. So E1A stimulates the cell cycle by releasing PRB, but also would be normally stimulating apoptosis if it didn't also have these E1B proteins. Turns out that this E1B protein, together with E4, which is not as critical, leads to more ubiquitination of P53. Some of you may recognize this protein, culin. Culin's a protein and ubiquitin ligase that Dr. Singer's lab works with here down at the CLSB. So I'll let him tell you all about that. So that's what E1A, E1B are doing. So that's regulating the cell cycle again, turning on replication, blocking apoptosis. It's great, but you still need to replicate your genome. What are these other E2 proteins doing? Well, they're these terminal proteins, DNA polymerases, and DNA binding proteins, which are present. So this is the genome as it's released. Then you make the E2 proteins, including DNA polymerase, this pre-terminal protein, and single-stranded DNA binding protein. The pre-terminal protein has a cytosine residue which gets attached to it on an OH, but unlike the case you have with VPG, which attaches it to a, what kind of amino acid? Tyrosine. This is a serine. So there are really only three possibilities. You could have tyrosine, serine, or threonine in terms of OHs. Here it's just serine, which is being used. So serine then attaches to this cytidine. Then that will base pair with the guanosine residue, which is at the 3' end of your antigenome, and the DNA polymerase, which is a viral encoded DNA polymerase. It's not the cellular DNA polymerase, because cellular DNA polymerase is like RNA primers, and they've got specific origins that they interact with. They also need helicases, they need all kinds of other stuff. Here, it's just the viral DNA polymerase that interacts with this primer on the terminal protein. It will replicate the genome all the way down from one end to the other. At the same time, the viral single-stranded DNA binding protein is binding to that opposite strand. Just displaces that as it moves along down to the other end. So we end up with one new copy and one old copy, and then this strand which has been displaced and has all of this single-stranded DNA binding protein attached to it. This is where those inverted terminal repeats come in. Mentioned before, they can also base pair with the opposite end, so it can base pair with itself. This is that strand which was just displaced that has a nice guanosine residue here, the cytidine residue that's attached to the serine of another pre-terminal protein can bind to, and the DNA polymerase can replicate its way around this particular genome. One question you may be asking is, well, okay, at all of the eukaryotic origins of replication, you need a helicase, right? Here you don't seem to need one. Why don't you need one? Well, it turns out that this single-stranded DNA binding protein binds to single-stranded DNA so well that it basically serves as the helicase. So the affinity of the single-stranded binding protein for single-stranded DNA is so high that it's basically pulling the two strands apart as they're being replicated by the viral DNA polymerase. So we've gotten into the cell, we've turned on the cell cycle, we've made more of our genome. What do we need now? Late proteins. So how do we make our late proteins? The major late promoter is here. Again, all of the late proteins, every single one of those structural proteins that ends up going into the virion is made from this one promoter, which has three different parts which are always getting spliced together at the beginning here. This is what Robertson Sharp showed for the hexon gene, which is over here in this one segment. But each of these gets spliced together, splice those together, and then splice to either this set, this splice acceptor site, this splice acceptor site, etc. So you end up with a set of different RNAs that then get differentially spliced as well. Here's just the example of what happens in L1. 3 different possible splice sites that can happen here, giving you 3 different RNAs. Most of these give you a different ATG in terms of making a different protein. Same thing happens over here with your L3. All of these, L1 through L5, have to do with where you have CSTF binding to and cleaving that RNA as it's being made. And a lot of the discoveries in terms of RNA transcriptional termination, where you get poly-A tails, was not surprisingly found from the study of the adenovirus major late promoter. Why do you think having this 5' end be identical for all of these things would be really useful? It's right here underneath it. So transport and translation. So getting these RNAs out of the nucleus and getting them translated. Translation, turns out that this tripartite leader is really good in terms of translational regulation. You get a lot of all of these genes here that are made in terms of structures and putting all of them together. So you've made all of your proteins. They then get assembled onto your genome. You're ready to go and get outside the cell. But remember, this is a naked virus. It's not nice and enveloped. You can't go out and bud at the plasma membrane. You've got to get out some other way. And so that seems to require the death protein. Most important is these here. These are proteins which are made from early promoters, but it turns out that it just takes a long time to get enough of them. This is not unlike accumulation of lysozyme, accumulation of holins that you get in some of the bacteriophage. Exactly how it works is not clear here. It's not apoptosis, because apoptosis is breaking everything down. This basically seems to put everything in stasis until you assemble enough virus particles for them to get out. There are a couple of other proteins here. E4, which kills cancer cells in a p53 independent fashion. Again, this is probably important for the whole death process here. And remember, we always try to knock out apoptosis. We don't want apoptosis here. That's what all of your E1 and particularly the E1B proteins are involved in. So people have been studying adenoviruses literally for decades, and part of that has led to now using what we've learned from a lot of these viruses to try and develop some therapies. So gene therapy, as I mentioned before, these are really nice gene therapy tools because they get into the cell nicely, they get transported to the nucleus, you have your DNA which is there. Turns out that having a linear genome is really pretty flexible. You can have larger or smaller amounts, put extra pieces of DNA in there. It doesn't make a huge difference. Of course, you don't want these viruses replicating, and you don't want them leading to tumorous development. So almost all of these gene therapy vectors are lacking in E1A and E2A. So they don't have those proteins that are regulating p53. What's interesting is some other people kind of took this idea and flipped it on its head and said, well, if we don't put in E1B, which is degrading p53, it's shutting down apoptosis, we make an adenovirus that doesn't have E1B in it, but still has E1A, it's going to be trying to make more virus, and it's going to be stimulating p53, the way we talked about before, leading to not just progression through the cell cycle, but also eventually apoptosis. These are really neat because they should be completely inactive in a cell that has normal p53. Normal p53, you don't regulate it, you're going to have a rest in the cell cycle, the virus is not going to be able to replicate. If it does, then it goes to apoptosis, you have no problem. On the other hand, if you have a cell that doesn't have p53 in it, this should allow virus replication to make all of those death proteins and just kill off cancer cells. So in theory, an absolutely wonderful idea, but the company went bankrupt about 15 years ago. So, well, not quite, 12 years ago now. So that's what's happening, or was happening, with this whole idea of tumor therapy. People are thinking about using this as well, but in a slightly different form, and we'll talk about that right at the end. More recently, there's been a whole bunch of press on the fat virus, Adenovirus 36. Some of you may or may not have heard about this. In mice, infection with Adenovirus 36 caused this big increase in adipose tissue. A couple of references here. 5 years after that, Adenovirus 36 antibodies were found in humans, 30% in obese humans, 11% in non-obese humans. Wow, looks like a big difference. Adipose-derived stem cells are stimulated by infection by Adenovirus 36. These rat cells only by Adenovirus 4 or Adenovirus 36. We could look at this Fox News thing. It's all about how this nasty virus, and so if we can control virus infection, we can control obesity. Isn't it going to be a wonderful thing? But you can see my comment down there at the bottom. So turns out that almost all of these were published by the same people who started to publish in less and less, so maybe, I'm not sure what the right term is here, internationally recognized journals. And probably most recently, there was this study in 2010 where 78% of the kids were adenovirus positive and obese, and 7% were adenovirus positive and not obese. But if you look at the numbers here, these numbers are ridiculously small. And there's a really nice discussion in TWIV 18 and 101 about why this stuff is really probably garbage, because you find adenoviruses all over the place. So adenovirus usually isn't a problem in terms of probably not obesity either, but certainly not much for most respiratory diseases. But there are some differences to that. And the main one, is also mentioned in the textbook, is that adenovirus type 4 and adenovirus type 7 cause really nasty respiratory diseases in a bunch of military recruits. And so, I don't know, how many of you have been in the military? Not me. So, very close quarters in basic training, very little ways of controlling respiratory diseases, things that are getting around there. So adenovirus 4 and adenovirus 7 were originally found because they were causing lots of disease in military recruits. One of the things that really amazed me is that the vaccination process is 2 pills. Now, how do you use pills to treat a respiratory disease? It's like, how the heck is that supposed to work? Well, apparently some of these viruses, and particularly adenovirus 5 and 41, actually replicate in the gut, which was a surprise to me before I put this together. But these adenovirus 4 and adenovirus 7, if they don't go in respiratorily and they go in the gut, they can actually give a nice immune response. And so just this last year, actually a couple of years ago now, they actually discontinued using this as a vaccine and then reinstituted, I guess it was 2011 or something like that, taking the pills, and their reduction in acute respiratory disease went down by 162%. So really amazing process of reduction here, and just by taking pills, and literally these pills, unfortunately I had an image here which I thought was saved, I guess it didn't get saved, where it's live adenovirus 4 and adenovirus 7, 1 pill of each, which you swallow, and then it gives you a nicer immune response to it. And I say I've just recently been re-added to basic training in terms of the immunizations that you get there. But partly because of this process, you can take these in pill forms. They're really stable because of this amazing structure that gets put together. They work in pills. You can get them down into the digestive tract. A lot of people are trying to use these now for developing vaccines, not to the adenoviruses, but to other proteins that have been added to the adenoviruses. Ebola is, of course, one obvious one that everyone's interested in. But I think more excitingly, they're actually trying to use these for flu. And one of the nice things about using a recombinant viral vaccine is you can put basically any antigen you want into this particular genome and have it express. You get a nice immune response to them. So at least in theory, this would be really nice. And the great thing about pills, they're really stable. So you can just have them on the shelf. Don't have to worry about keeping them cool, fridge temperatures, etc. So a pretty exciting, I think, new area for vaccines. So I completely forgot to bring all of your Scantrons with me. I have them. I promise I will have them on Friday. They're in the folder in my office. If you want some of them, you can follow me over there right now. Otherwise, we'll talk about herpes viruses on Friday.